Thanks a lot for the kind introduction. And thanks also to the organizers for giving me the opportunity to talk today about our paper that we recently published. Today, I would like to talk about our work on Rorgt disease and their role in inducing peripheral Tregs in response to oral antigens. But before digging into the data, I would like to give you just a brief introduction in what exactly dendritic cells are. So dendritic cells can be subdivided in two major fractions. One of the dendritic cell fractions is called conventional DCs, and the CDC1 compartment was shown to be detrimental in inducing T cell responses, which will fight tumors by priming cytotoxic CD8 T cells, or by actually priming T helper 1 cells, which will actually be formed due to intracellular pathogen insult. On the other side, you have the CDC2 compartment, which was shown to be detrimental to prime the Th17 or the Th2 compartment. And these immune responses are normally built against parasites, extracellular pathogens, or in certain conditions like autoimmunity against allergens. In the recent years, there were also found a novel subset of antigen-presenting cells. These APCs were defined by the expression of Rorgt and were shown to be detrimental to induce peripheral Treg induction against commensal bacteria. So the Rorgt APCs, they were actually subdivided in two major fractions. One of the fraction is basically the Rorgt expressing ILC3s, and then you have also the Rorgt-positive DC cell subsets, which were termed also in different ways, like, for example, fetus cells, which were named by the group of Brown, the tollogenic DCs, which were named by the group of Littman, as well as the Jano cells or the ETEC cells. Just for the simplicity, whenever I'm referring to the Thetis cells or toll DCs, I always use the nomenclature we gave them, which is the Rorgt DCs. So how I mentioned already in the beginning, the Rorgt APCs were shown to be very important to mount a tolerogenic immune response against commensals. 3 seminal papers were published back-to-back in Nature in 2022 by the group of Dan Littman, as well as Sonnenberg, and as well as Chrissy Brown. However, one open question which remains is whether Rorgt APCs are also important in inducing and promoting tolerance to food antigens. Obviously, there is no good model which affects the development of these Rorgt APCs, and this was basically the starting point on when I started to work on this project. So my question was, can we generate a mouse line that will specifically affect the Rorgt APCs? And to solve this issue, what I did was actually to have a look on publicly available ATAC data sequencing data sets. And what I realized is that if you look at the ILC3 peaks, you can clearly detect an open chromatin region within a 7 kb downstream of the start codon of the Rorgt isoform. You can also see that the T cell compartment had no accessibility at all. So after have seen that and discussed with Marco, we decided actually to knock out this enhancer by flanking it by 2 guide RNAs. What I also realized, and sorry I forgot to mention, is that within this enhancer, we were able to find AHR and RUNX transcription factor binding motifs, which are known to be very important for ILC3 development. So we generated this enhancer knockout, and we call it from here on RORC plus 7 kb. And when we had backcrossed this enhancer knockout to homozygosity, the first thing we did was actually to investigate whether we had a phenotype in the ILC3 compartment in the small intestine lamina propria. So what you can see here in the wild-type littomate control, we have a very nice population of ILC3s, which is identified by the expression of Rorgt. However, if we have a look on our plus 7 kb enhancer knockout, you can clearly see that the ILC3 compartment is highly reduced. We also assess that obviously by frequency as well as total cell number, and you can clearly detect that the ILC3 compartment is affected in the small intestine lamina propria. We also had a look on the 3 different subsets of ILC3s, which can be distinguished by the expression of NKp46 as well as CCR6, and we realized that all the 3 subsets were actually reduced. Obviously, since Rorγt is a very important transcription factor, not only for ILC3 development, but also for the T cell compartment, we were wondering whether the T cell compartment might also be affected. So we had a look on the PTRACs, which are identified as CD4 T cells, which co-express Rorγt as well as FOXP3, and we could clearly detect a nice population within the wild-type littomate controls. However, when we looked at our 7kb enhancer knockout, there was a dramatic reduction of the PTRAC compartment. What was even more astonishing is that if we focused now on the Rorγt FOXP3 double negative CD4 T cells and used GATA3, which is a transcription factor essential for Th2 responses, we saw that in the wild-type compartment we had very few Th2s. However, in the enhancer knockout, there was a very big increase of the Th2 compartment. We also validated that by looking at the frequency of live cells as well as the total cell numbers. You can clearly see that the PTRAC is affected, so highly downregulated, whereas the Th2 is increased in our enhancer knockout mice. So obviously, after seeing that the Rorγt enhancer knockout has also an effect on the Rorγt PTRACs, we were wondering whether this T cell phenotype is due to a T cell intrinsic or driven by an extrinsic effect. So to assess this question, what we did was a bone marrow chimera. So we took wild-type bone marrow cells, depicted here as the black, in black, and as well as the enhancer knockout bone marrow cells, which are here depicted as the red population, and we co-injected that in a congenic recipient mouse line, let the bone marrows basically develop into the immune cell compartment and analyzed after 2 months. And what you can clearly see is that if you gate on the ILC3 compartment, the majority of ILC3s in the mesenteric lymph nodes, as well as in the small intestine lamina propria, were mostly derived from the wild-type compartment. And very little was actually from the RORC plus 7kb enhancer knockout. However, when we now checked for Rorγt expressing T cells, we could clearly see that they're formed by both the wild type and by the enhancer knockout mouse. And this was true for the mesenteric lymph node as well as for the small intestine lamina propria. This already indicates that the RORC enhancer is actually very potent in inducing the development of ILC3s, but seems to be negligible for the development of PTRACs or Th17, which both require Rorγt. So after have seen these results, we were wondering whether the lack of the PTRACs could induce some kind of low-grade inflammation. So to assess this question, what we did was to measure the levels of lipocalin, which is basically a protein which is secreted upon inflammation in the colon as well as the small intestine. When we actually compared the lipocalin levels in our wild-type mice to the levels observed in the enhancer knockout, we could clearly see that we had actually an increase in our enhancer knockouts. We also had a look in the gut permeability of in our mice, and for that we did actually a fitsidextran experiment by gavaging wild-type or enhancer knockouts, and also there we were able to see that the fitsidextran levels were increased in the enhancer knockout, which is a sign for increased gut permeability. So after seeing now that we have also a low-grade inflammation in our enhancer knockout, we are still wondering what the expansion of the Th2 could be driven by. Is it actually the microbiome or a disbalance of the microbiome which induces this Th2 response, or could it be that it's actually food-derived antigens which promote this allergic Th2 response? To assess this question, what we did first was actually try to kill the microbiome by treating our mice with antibiotics. So we took our wild-type mice as well as our 7kb enhancer knockout and treated these mice with an antibiotic regimen for 4 weeks. After 4 weeks, we analyzed the compartment and checked whether there was a difference in the Th2 and the PTRAC. As expected, and this was shown already in pretty many papers, if you take wild-type mice and you put them under antibiotic treatment for a long time period, you actually lack the PTRAC compartment. However, if we checked our enhancer knockouts, we saw that the overall decrease of the PTRACs was even higher if we treated our enhancer knockouts with the antibiotics, also depicted here in this summary slide. For the Th2 compartment, we're actually very happy to see that whenever you treated the wild-type mice with antibiotics, you saw that the Th2 compartment increased. Whereas in the enhancer knockout, we actually didn't see a difference in the Th2 compartment, which indicates that it seems even though you delete more microbiota, the Th2 expansion does not seem to increase. Obviously, the antibiotic treatment does not really abrogate and kill all the microbiome, but we still see that the Th2 response in the enhancer knockout mouse is increased. So we were wondering whether if we use a mouse which really never had contact with any microbiota species, whether these also have a Th2 increase. And for that, we assessed germ-free mice. So in germ-free mice, we basically saw that we completely lacked the peripheral Treg compartment. This was also already shown. And to our surprise, we also detected this increase of the Th2 compartment. So now take in mind and remember that these mice don't have any microbiota. So this Th2 expansion is most likely derived by another factor than the microbiome. So if I go now to the summary slide, what you can see here in the wild-type condition is that you have the IC3s, you have the T-cell compartment as well as Treg compartment. So whenever you have a wild-type condition, the Treg compartment is able to modulate immune suppressive environment, which keeps basically the inflammation in the gut intact. However, whenever you have a reduction of the Rorγt APCs, such as the ILC3s, you cumulatively have also a reduction of the Rorγt PTregs. This reduction will actually induce a reduced suppressive immune response, which leads also to a higher low-grade inflammation in the gut. For the Th2 expansion, it seems like that whenever you have this inflammatory condition, and you might also promote actually the proliferation of Th2 cells. So after having seen now all these results, we were wondering whether our ROS-C plus 7kb enhancer knockout mice are actually more susceptible to food antigen-based mouse models. For that, what we did was a very simple experiment. We saw purified CD45.1 OT2 mice and injected them into either wild type or 7kb enhancer knockout mice. Then we gavaged the mice with ovalbumin, and after 10 days, we analyzed the output of the OT2s in the wild type as well as in the enhancer knockout. And as you can see, whenever you have the OT2s injected into the wild type mouse, you are able to generate the Treg compartment. However, when you do the same experiment in 7kb enhancer knockout, you completely abrogate the priming of the peripheral Treg compartment. We also checked for the Th2 as well as for the Th17 compartment, and you can clearly see that the Th2 compartment actually was skewed in our enhancer knockouts, whereas the Th17 seemed not to be different compared to the wild type. So obviously, this is not the best model to check for oral tolerance. And to actually assess this question, what we took advantage of is a so-called allergic lung inflammation mouse model. This mouse model is very simple. So you take wild-type mice as well as 7kb enhancer knockout mice, and you tolerize them either with ovalbumin, which will be basically the tolerization against ova, or you can also gavage the mice with PBS. After a certain time, you immunize them with ovalbumin and alum, and then you re-challenge them by administering it intranasally. And then you check the lung and see whether there was actually a Treg induction or a Th2 response, more abundant. As you can see in our wild-type mouse controls, whenever you actually immunize the mice with ovalbumin, the Th2 response is actually kept under control. However, if you just immunize the mice, if you just tolerize the mice with PBS, you actually mount a very strong Th2 response. If you do the same experiment in our enhancer knockouts, the tolerization process with ova is actually not working at all. So you still mount a response and then a proliferative Th2 phenotype. We also had a look on eosinophils, and again, whenever you tolerize the mice with ovalbumin and the wild-type mice with ovalbumin, you can clearly see that the eosinophil levels were highly decreased compared with the tolerization with PBS. However, this tolerization process is completely abrogated in our 7kb enhancer knockout mice. We also assessed this inflammation by having a look on the leukocyte infiltration, and you can see again that in the wild-type mice, whenever you tolerize them with OVA, you have actually a negligible amount of leukocyte that infiltrate the lung. When you do the same experiment in wild-type mice with PBS, you have a massive infiltration of leukocytes. However, in enhancer knockout mice, in both conditions, in the PBS as well as in the over-tolerized model, you have this infiltration of leukocytes. We also had a look on over IgE antibodies, and we could clearly see that whenever in wild-type mice, whenever you actually tolerize them with ovalbumin, the over IgE response was highly decreased. However, this tolerization mechanism did not happen in the Rorg+ plus 7kb enhancer knockout. So obviously, we checked whether this 7kb enhancer is important for the ILC3 development, but we don't really know whether the other counterpart of Rorg-T APCs, which are the Rorg-T DCs, are also affected in the Rorg+ plus 7kb enhancer knockout. So to assess that, we took advantage of the data set which was published by the group of Brown in the Nature paper, and we had a look on the Tethys cells, which are basically the Rorg-T DCs, and checked whether the 7kb element is actually accessible in the Rorg-T DCs. And indeed, you can clearly see that the Tethys cell compartment has also open chromatin within the 7kb region. So how can we visualize these cells, to prove the point that they are actually affected by this 7kb enhancer? So to answer this question, we actually took advantage of GM38411. This is an antisense long non-coding RNA that uses the same promoter as the Rorg-T isoform. And what we did was actually to generate an iCRE human CD2 reporter mouse. And we are hoping that with this reporter mouse, human CD2 expression would actually give us an indication for the actual Rorg-T expression. And indeed, when we actually analyze the small intestine lamina propria as well as the mesenteric lymph node and look for human CD2 positive cells, which is basically the surrogate marker or reporter construct for GM38411, you can clearly see that the ILC3 compartment highly expresses human CD2. What we also realized is that within the migratory DC2 compartment, there was a small fraction of cells which also expressed the human CD2 construct. Obviously, we were wondering whether these are actually the Rorg CDCs. And to assess this question, what we did was to perform single-cell RNA sequencing from our reporter mice. So we did actually harvest small intestine lamina propria as well as the mesenteric lymph node compartment, and we were able to detect a very tiny population of Rorg-T DCs. As you can see, they actually share a lot of genes which are similar to the dendritic cells, such as DPP4, ZBTB46, CD11C, and MHC2, but they have also expression of genes which are known to be very specific for the Rorg CDCs, like, for example, RORC, PRDM16, as well as IL-18 receptor 1 and CCR6. To basically show that our reporter is actually reporting the same way as the RORC reporter, for example, we also showed and highlight the expression levels of RORC and compared to the expression level of the GM38411 or our human CD2 reporter. And you can clearly see that it's always within our RORC CDC cluster here where we have this expression. But obviously, we were wondering, since we have such a tiny population, whether we are able to enrich for the Rorg-T DCs by taking advantage of our reporter mouse line. So what we did was actually to sort purify from 12, 15 days old pups, the mesenteric lymph node compartment, which was human CD2, so Rorg-T positive. And then we performed, from all the cells we obtained, a single-cell RNA sequencing. And what you can see is that indeed, we are able to detect actually 4 subsets of Rorg-T DCs. We had a look on the expression levels, and you can clearly see that these 4 subsets of Rorg CDCs express RORC. They express our long non-coding RNA GM38411, as well as the human CD2 construct. At the same time, they express PRDM16, as well as markers which were shown to subdivide the different Rorg-T DC subsets like CCR6 as well as AIR. We also compared these 4 different clusters to the clusters which are already identified and characterized by the group of Brown in 2022. And we can clearly see that the Rorg-T DC clusters we found are equivalent to the clusters that Brown identified in her Nature paper. So obviously now the next question is, does the 7kb enhancer knockout regulate and abrogate development of Rorg-T DCs? So for that, what we did was, again, a single-cell RNA sequencing on wild type as well as Rorg-C enhancer knockout mice. And when we now focus specifically on the raw gamma TDCs, which is indicated here as cluster 8, you can clearly see that the majority of raw CDCs are actually derived from the wild-type counterpart and not from the knockout. We also validated this experiment by having a look on the frequency of the raw gamma TDCs as well as the total cell numbers, and we were able to show that indeed the enhancer knockouts seem to have a reduction of the raw gamma TDC compartment. So taken together, what we can actually say is that in the wild-type counterpart, you also find raw gamma TDCs, which might also be important for the priming of the PTRAC compartment. So whenever you are able to induce this tolerogenic immune cell subset, you will have also an immune suppressive action towards the Th2 compartment. However, in our Rorg-C plus 7kb enhancer knockout, you have a reduction of the ILC3s as well as of the raw gamma TDCs, which is going along with a reduction of the PTRAC compartment. The reduced PTRAC compartment will actually induce a lower suppressive immune response, which leads to an expansion of this Th2 compartment. But the next question is, which subset is specifically priming the PTRACs, right? Because in our 7kb enhancer, we have a reduction in the ILC3 compartment, but we have also a reduction in the raw gamma TDCs. So to assess this question, we basically generated a new mouse model, which will specifically target the ILC3 compartment. The gene we targeted for this purpose was serine-C2, and we integrated at the 5' UTR an iris ICRE cassette and crossed these mice then to the rosa tomato mice. And what you can see here is that indeed the ILC3 compartment is almost all labeled within the serine-C2 compartment. You have also very low labeling within the group 1 ILCs or the ILC2s. Astonishingly is that you lack actually a big fraction of the T cells, which normally is labeled if you use, for example, the raw gamma T-CRE. We also checked for raw gamma TDCs, and we saw that the raw gamma TDC compartment is not affected at all by the serine-C2 CRE. So we were wondering, can we now cross the serine-C2 mice to Rorg-C flox mice and generate a mouse that completely lacks the ILC3 compartment? And that's what we did. So we intercrossed the serine-C2 with the Rorg-C flox and then assessed the frequency of ILC3s in the small intestine lamina propria as well as the mesenteric lymph node. And what we saw is that if we gate now on B220 negative, CD3 negative cells and basically gate on CD45 positive as well as Ty1.2 positive cells, you can see that in the litomate controls, which are CRE negative, you have the ILC1 compartment as well as the ILC3s, which are also described as being a little bit lower for CD45. However, in our serine-C2 mice, which are CRE positive, you completely lack this population. We further validated that by gating now and specifically use GATA3 as well as raw gamma T, and you can clearly see that the ILC3 compartment, highly abundant in the wild type, is completely lacked and not present in the serine-C2 CRE mice. Now the question was like, do these mice still prime PTRACs and inhibit the expansion of T cells? So what we did was actually to assess the PTRAC compartment, and we were very interested to see and happy to see that the PTRACs in our CRE positive mice were not affected at all. And this is true for the frequency as well as for the total cell numbers. We also had a look on the Th2 compartment, and also there we didn't see an expansion as we saw with the 7kb enhancer knockout. So taken together, in our ILC3 specific depleter mouse model, we lack the ILC3s, but we still maintain the priming of the peripheral Tregs and the immune suppression towards the Th2 compartment. So another open question which remains is, where exactly do raw gamma TDCs come from? And as Javier was saying at the beginning, I'm very interested in the development of dendritic cells, so I tried to tackle this question as well in our paper. And I just want to give you a brief introduction and tell you that DCs, they're very heterogeneous and they have actually different sources of development. So they can be generated from the myeloid progenitor, which is known as CDP, but there are also a lot of papers showing that also CLPs or the IL-7 receptor lymphoid progenitors have the potential to generate conventional DCs. So knowing this, I was actually wondering whether the raw gamma TDCs derive from lymphoid or myeloid progenitors. And to assess this question, we took advantage of the human CD2 Cree mice, which are backcrossed to the rosa tomato. So the human CD2 Cree mice are known to be very specific and lineage tracing only the lymphoid compartment. So we had a look and gated on a CCR6 positive Rorγt disease, and we were very surprised to see that actually the majority of the Rorγt disease were lineage traced in our human CD2 Cree mice. So knowing that actually FLT3 ligand injections induces the proliferation not only of dendritic cells, but also lymphocyte progenitors, we wonder whether if we would inject 15 days old pups with FLT3 ligand, whether we would expand the Rorγt DC compartment. And we were very happy to see that as a bulk, the Rorγt DCs were highly increased in cell numbers when we injected the FLT3 ligand into the pups. However, the only subset which seems not to be dependent on FLT3 ligand injections was actually the Rorγt DC1 subset. However, the subsets 2, 3, and 4 reacted all to the injection of FLT3 ligand. As a positive control, we also had a look on the ILC3 compartment as well as the group 1 ILCs, and you can see that they all also increased upon injection of FLT3 ligand. Knowing now that FLT3 ligand can expand these cells, we were wondering whether we can generate the Rorγt DCs also in vitro. And the first thing I did was actually to test whether total bone marrow cells, when cultured under FLT3 ligand or cultured under lymphocyte priming conditions such as FLT3 ligand and IL-7 and stem cell factor, would induce the generation of Rorγt DCs. And indeed, even though in a very, very low frequency, but whenever you cultured total bone marrow cells from 15 days old mice and added FLT3 ligand, you got a very, very small fraction of Rorγt DCs within the CDC2 gates. However, if you added stem cell factor, IL-7, or stem cell factor and IL-7 combined, you could gradually expand these populations, and this is also observable here by this cumulative slide. Obviously, the next question is, we can generate the Rorγt DCs from total bone marrow DC cultures, but can we also do the same by sort purify specific progenitors and put them in culture? And what we specifically did was to sort purify lymphoid as well as mild progenitors, and we checked whether under these conditions they were able to generate the Rorγt DCs. So whenever we sorted the LMPPs, which is one of the earliest progenitors in hematopoiesis, you could clearly detect that the LMPPs were very good at generating the Rorγt DCs and gradually expanded whenever you added stem cell factor, IL-7, or stem cell factor and IL-7. However, if you sort purified now the lymphoid progenitor, you still saw this phenotype. So the IL-7 receptor positive lymphoid progenitors had the potential to generate these Rorγt DCs. However, when you did it with the myeloid progenitor, which is known to give rise to dendritic cells, the CDPs, they completely lacked, and they were not even able to expand the generation of Rorγt DCs in stem cell factor and IL-7 and FLT3 ligand conditions. So if we go back to the summary slides of this hematopoietic tree, what we can say is that Rorγt DCs seem to be able to be generated by IL-7 receptor positive lymphoid progenitors. However, the myeloid progenitors, the CDPs, seem not to have the potential to do so. And with that, I actually finish my talk. I just want to also highlight a little bit all the papers which came out this year. I mean, the field is exploding, and if you are interested about this topic, please go back. There are beautiful papers published by the group of Daniel Musida, Barbara Schrammel, our group, Dan Littman's group, or James Gardner's group, Chrissy Brown's group, as well as Katmi's group. So go into that and have a look on those papers. And with that, I want to acknowledge everyone in the lab, especially Marco, who gave me the possibility to work on such an exciting